Get started. Greetings from the land of the Darg and the Gundagar, where we pay respect to the traditional custodians of the country on which we meet. My name is Kevin Joseph, and with me here is Hamish Clark, and we are the organizers of Science at the Local. Science at the Local is a community initiative in the Blue Mountains that's supported by Inspiring Australia, which is a Commonwealth government-funded program. So the focus for Hamish and I in Science at the Local is to strengthen the bonds between local communities and scientific research. So we run various engagements and events designed around building community through science. So as we take our steps towards the new normal, I hope that you will consider joining us in person early next year for our events in Springwood in the Blue Mountains. So you can see what we're up to and keep track of our events through Facebook, our website, or you can even send us an email and we'll add you to our email list. So I will throw you over to Hamish to introduce our speaker for today's webinar. Hamish.
Thanks very much, Kevin. Great to see you all virtually. We've got 29 people in the room today, so welcome to all of you. It's great to have you here. Thanks for dialing in. So it gives me great pleasure to welcome our speaker today, Professor Michael Manefield. He's a co-founder and former director of another science communication series called JAMS, Joint Academic Microbiology Seminars. He's also the author of a series of illustrated children's books on microbiology and the founder of a company, Novorem, supporting industry with environmental microbiology expertise. He holds a professorial appointment in the School of Civil and Environmental Engineering at the University of New South Wales. He's teaching environmental science and engineering and researching pollutant biodegradation, biogas production, isoprene metabolism and wastewater treatment. Would you please join me in making Professor Michael Manefield very welcome? Over to you, Michael.
All right, thanks, Hamish. Can you guys see my slides?
Looks good, yep.
Awesome. All right. Hi, everybody. This is a little bit weird. I can't see anyone. I'm kind of used to that, but I don't know any of you. And I don't know what your background is. And so I'll be doing my best to share some of the research that we've been doing at the University of New South Wales to a general audience, but also, you know, make it quite sciencey, so you feel like you've had a dose of science as well. So I want to talk to you for the next half hour about bioremediation. So it's the use of microorganisms to degrade contaminants in the environment. And specifically what I wanted to do is present some of the work we've been doing under a New South Wales Environment Trust grant on dioxin biodegradation in the Sydney Harbour. So I want to acknowledge as well my collaborators, Keith Osborne and Casey O'Farrell, so government and industry represented, and Dr. Matthew Lee, who I've been working with, have been in research partnership for the last 15 years, a lot of great work together, and the master's student who was working on this project, Gan Liang, who starting from scratch, did a pretty amazing job with this research. So a bit of a quick sort of background introduction to microbiology. On the left here, you see the sort of tree of life as I was taught it in high school. It's very picturesque and pretty, but it doesn't really show you much of the microbial side of life. On the right, we have a different representation, actually drawn as a tree, which is quite cute. And what this shows you is that there are three main branches to the tree of life. And we're sitting on the tip of this one here. But it also, what it's basically showing you in this picture is that there's two of the main branches out of the three are all microbial. So there's the bacterial branch. Most of us have heard of that. There's the archaeal branch. Many people haven't heard of the archaea. And they're only really recognized as being on a very distinct trajectory in evolutionary history of life on Earth in the 1980s. And here's the eukaryotic branch, which is what we sit on, along with all the other animals and all the plants and all the fungi and the protozoa. So essentially, the tree that you see on the left is really just the tip of this branch here. It's missing all of this bit. And my main point here is that most of the biological diversity on Earth is actually microbial or microorganisms. This is what the tree of life kind of looks like today through phylogenetics, so sequencing DNA sequences and figuring out relationships based on similarity or differences between those sequences. You see that the bacteria are the main source of diversity in life on Earth. And here we have the archaea and the eukaryotes, which are actually quite closely related. All right, but if diversity doesn't float your boat and you're not impressed by that, let's do it by numbers. So the number of microbes on Earth is estimated to be around 10 to the 30. For those of you not familiar with scientific notation, that's a 1, 30 zeros. And we can compare that to something like the number of stars in the universe. So there are around 100 million microorganisms on this planet for every star in the universe. We're talking some very big numbers. Some other numbers there on the screen for comparison. Number of human cells in your body, it's around 10 to the 13. Number of human cells on the planet, multiplying those in your body by 7.5 billion, we get to around 10 to the 23. Human cells are hopelessly outnumbered by microbial cells by many orders of magnitude. But you might think to yourself, yeah, but human cells are actually bigger than microbial cells. But even if we generously account for the difference in cell mass between microbial cells and human cells, for every kilogram of human on the planet, there's 100 tons of microorganisms on the planet. So it becomes easier to sort of believe that microbes drive the biogeochemical cycles of the planet. So I'm not sure if this is familiar to anybody. This is a brilliant image from a film called Princess Mononoke out of Studio Ghibli in Japan. I'm a big fan of these films and I've watched this one many times. Many of them have a theme of conflict between humankind and nature. And that's true of Princess Mononoke. She's my kind of princess, as distinct from a Disney style princess. She's a bit of a warrior princess. But in this film, there's a character called the forest spirit, or the spirit of the forest. And it rises up out of the forest at dusk and sort of causes things to be fertile and causes things to decay, and in essence drives the geochemical cycles of the forest or of nature. And I was sitting there watching this film at some point, probably in the 5th viewing or something. And all of a sudden I realized that that creature, that spirit of the forest that rises up in semi-humanoid form with its head in the clouds, is more or less symbolic of the microbiology of the forest. So what we're dealing with here are invisible self-replicating catalytic fat bubbles that are governing the geochemical cycles or the ecosystem services of the Earth by virtue both of their diversity, which I've described, and their staggering abundance. So in some level, the Earth does have a spirit. It's just doesn't rise up like it does in Princess Mononoke. It's made of microorganisms, 10 to the 30 of them in the oceans, in the soil, in the atmosphere, in us, on us, everywhere. All right, so on to the main topic of today's talk. I'm going to talk about Sydney Harbour, this very iconic, picturesque scene that we're looking at here. If you're in the Blue Mountains, you can probably see yourself in the background. You can wave, hi. So I developed an interest in some of the contaminants in Sydney Harbour, and I'll explain why in a minute. But this pristine, picturesque view hides a dirty secret in the big, shitty, pretty city. That's from a Skunk Hour song, if any of you remember Skunk Hour from the 90s. So on around Homebush Bay, on the Rhodes Peninsula, there was a lot of manufacturing activity, and certainly around the harbour and Parramatta River, there's a history of manufacturing activity and pretty intense chemicals production during the 1960s on the Rhodes Peninsula by a company called Union Carbide, which no longer exists, now owned, I think, by Dow Chemicals. And they were producing herbicides and pesticides, dichlorophenoxyacetic acid and trichlorophenoxyacetic acid on that site. And it was part of the Vietnam War effort that they were producing these herbicides in the form of Agent Orange. And these compounds in themselves aren't particularly great chemistry to have around, but the preparation was contaminated. So it was just a byproduct of the manufacturing by chemicals called dibenzodioxins. And so that site ended up quite severely contaminated. So waste was being used to reclaim land. And so Homebush Bay became, and the sediments in Homebush Bay became really quite intensely contaminated. And remediation works have removed the bulk of the contaminant mass out of sediments of Homebush Bay, like the really steaming stuff, the really hot stuff. But a residual legacy remains in the harbour sediments, and that continues to distribute dioxins throughout the harbour. So the dioxins stick to really tiny particles about the size of bacteria, which is about a micrometer or 100 times smaller than the width of a human hair. And so we have Homebush Bay serving as what we call a source zone, distributing dioxins throughout the harbour in perpetuity. So these compounds are not only pretty nasty, but they are very recalcitrant or resist degradation in the environment. So a very slow breakdown. So when we talk about dioxins, it's actually an umbrella term for a whole bunch of different chemicals. And in that, under that umbrella are the chlorinated or polychlorinated dibenzodioxins. So you can see here just the basic dioxin structure, just this ring structure with 2 oxygen molecules. Then there's dibenzodioxin, these 3-ring structure on the right-hand side of the slide. But I want to talk briefly about the health impacts of one of the particular specific compounds called 2,3,7,8 tetrachlorodibenzodioxin, which I will refer to as TCDD from here on in. And it's shown more or less in the middle of the slide, the 3-ring structure characteristic of the dibenzodioxins, and then these 4 chlorine atoms. That's why it's a tetrachlorodibenzodioxin. So they are natural products. They're naturally occurring through combustion. So anytime you burn something, you will produce dioxins. So bushfires produce dioxins. But they've been produced and released in higher concentrations through chemical manufacturing of byproducts. So dioxins activate what's called the aryl hydrocarbon receptor in our bodies, and this impacts gene expression broadly, and it results in stillbirths and birth defects. And dioxins are also classified as carcinogenic, so cancer causing. And the health impacts of dioxins, which were spread all around Vietnam as part of the Vietnam War with Agent Orange, a contaminant of Agent Orange, serving as a defoliant for the wilderness in Vietnam. The health impacts of dioxins that were delivered in Agent Orange, synthesized on the Rose Peninsula in Sydney, are depicted graphically in the War Remnants Museum. I don't know if anyone's ever been there. It's in Ho Chi Minh City in Vietnam. I had a PhD student in my team who was from Vietnam, and once she'd finished her candidature, I went over and visited her in Can Tho down in the Delta and gave a presentation. And then we went doing some touring around Ho Chi Minh City. And we went to this museum, and I saw all these amazing pictures of people who have birth defects as a consequence of exposure to Agent Orange, or specifically the byproduct dioxin in the Agent Orange. And I was really moved by it. I basically stood in the museum and cried, and decided that I would like to start doing some research on the biodegradation of these compounds, because we were doing a lot of research on related chlorinated compounds, and we thought maybe we can actually do something about this. So questions that we sought to address in the study once we got funding from New South Wales Environment Trust are fourfold. First one was, are dioxins still present at high concentrations in the harbour sediments? And is dioxin degradation occurring in the harbour sediments? Is there any evidence of that? Are there dioxin degrading bacteria in the sediments? And can we grow dioxin degrading bacteria from the sediments? And so I want to present to you some of the results of that research. The first thing we did was enlist a guy named Stuart Taylor, who has sadly since passed away, to take us out onto the harbour and do some coring of harbour sediments all around different locations. And so this map shows the location of 10 sampling sites. 4 of them were in Homebush Bay. This is the Rhodes Peninsula on the left. And then another 6 sampling locations sort of moving away from Homebush Bay. We took 10 50 centimetre core samples, and they were divided into 2 25 centimetre upper and lower fractions. And we quantified the concentration and composition of the dioxins in those samples. We extracted DNA to sequence that DNA to identify what microorganisms were in the sediment samples. And then we used sediment samples as inoculum or inocular for laboratory microcosms, little just basically trying to grow the bugs in flasks in the laboratory. So this is a graph showing the concentrations, the total concentrations of dibenzo dioxins and dibenzofurans in the harbour sediments. They're very much still there. And what we found was that most of what's in there is the octachlorodibenzodioxin congener. That's this dibenzodioxin structure with the 3 rings and 8 chlorine atoms adorning it. We found that the highest concentrations were in Homebush Bay, and also some of the lowest concentrations were in Homebush Bay, but there was no real trend in upper or lower core, and there was no real trend moving away from Homebush Bay. Certainly the highest concentrations are in Homebush Bay, but we were kind of expecting to see a gradient as we moved away, but it's, you know, the dioxins from Homebush Bay are more or less everywhere, or at least everywhere we sampled. So these 2 graphs are showing the upper and lower contaminant profiles of the cores over these 10 sampling sites, but it's all broken down into the different congeners, the different types of dibenzodioxins that we were actually quantifying. There are so many of them that we can't quantify all of them, but we quantified a pretty comprehensive set of them. And this data is presented in what we call HUTEC, a unit called HUTEX, which is the toxicology equivalent of the concentration. So these are not absolute concentrations. These are representing the toxic load of the different dioxins. So whilst octachlorodibenzodioxin, or OCDD, is by far the most abundant one in the harbour sediments, it's not the most toxic one. So the most toxic one is the TCDD, the tetrachlorodibenzodioxin. And it's present at relatively low concentrations, but by virtue of its toxicity, represents a large part of the toxic load. So the octa, hepta, and tetrachlorodibenzodioxin congeners make up most of the toxic load, even though the hepta and the tetra aren't particularly abundant, they're much more toxic. And so the toxic load due to the dioxins in Homebush Bay, it's roughly 400 times the global background of dioxins. As I mentioned, dioxins are naturally occurring, so there's some around everywhere, always. And the global background is sort of a concentration of around 5 picograms per gram of sediment or soil. In Sydney Harbour generally, at large, on average, the concentration is considerably higher than that, around 40 times higher at 200 picograms per gram. And in Homebush Bay, it's really quite nasty at 2,000, an average of 2,000 picograms per gram. So the problem is still there. So this particular slide might freak everyone out. It looks pretty complicated, but I don't want people to freak out too much. There's just a few things I want to point out here. I just want people to pay attention to the first congener on these graphs. 3 of these graphs, the 2 on the left and the 2 on the top, are just different samples within Homebush Bay. And the bottom right one is the average of those 3 samples. So, you know, more or less just presenting all the same data. And the black bars are data from an earlier sampling campaign that happened in 2003, published by Birch et al. In 2007. And the red data is from our sampling campaign. And what I want you to notice on all of these graphs, the first 2 bars, the first red one, the first black one, that's the tetrachlorodibenzodioxin, or the TCDD. And what you can see is that the black bar from 2003 is much bigger than the red bar from the more recent sampling campaign. So the tetrachlorodibenzoedioxin concentration in Homebush Bay has actually decreased quite significantly over that period of time. So it's more than halved in Homebush Bay. And so we also observed for the first time predicted breakdown products of the tetrachlorodibenzoedioxin. So trichloro, dichloro, monochloro dibenzodioxins, and they hadn't been seen before. So it seems as though naturally these compounds are breaking down in the sediments. People have projected that they'll be there for a very long time, and I have little doubt that that's true. But we were quite encouraged by the fact that the concentrations had been decreasing over a period of 10 to 15 years. Kind of exciting, yay nature. Question is, how is that happening, and is there anything we can do about accelerating it? So dechlorination, or the removal of chlorine atoms from dioxins by bacteria, that's been observed previously. So it's a known thing. Tetrachlorodibenzoedioxin has been shown to break down to trichlorodibenzoedioxin. This is a reductive process, if people can remember their redox chemistry. Reduction is the gain of electrons and oxidation is the loss of electrons. So if electrons can be delivered to these carbon to chlorine bonds in these molecules, it releases the chlorine atom. And so if you take off just one chlorine atom from tetrachlorodibenzoedioxin, you end up with a much less toxic compound. So you can actually, through dechlorination, reduce the toxic load. And I've just put a reference here by Bunge et al. Out of Leipzig in Germany. And that was the first observation of a dioxin dechlorinating bacterium. So this is something that's been seen before. That was published in 2003, almost 20 years ago now. So you know, we're not entirely surprised to see something like that going on in the Harbour sediments, but certainly pleased to see it. So to move on with this, I'm going to have to give you a little introduction to my favorite kind of bacterium. These are the organohalide respiring bacteria, or the ORB, or orb. And so these are fascinating creatures. I've got pictures of two of them here on this slide. One's called Helicocoides, that's on the top, and another one's called Helibacter. So a spherical one and a rod-shaped one. Two different phyla, the Chlorophyllaceae and Firmicutes phyla. And they have a quite extraordinary metabolism. So if you think of our metabolism in terms of a source of electrons and a sink of electrons, or our electron donor and electron acceptor, and life basically harvests energy from the transfer of electrons from our donor to our acceptor. For me this morning, for breakfast, my electron donor was Vegemite toast. And I am an obligate aerobe, so my electron acceptor is always oxygen. And bacteria are much more versatile than humans are in terms of what they can use for electron donors and what they can use for electron acceptors. And what these bacteria do, these organohalide respiring bacteria, they use hydrogen as an electron donor. That's what they have for breakfast. And they use chlorinated organics as their electron acceptor. So instead of respiring oxygen, they're respiring pollutants like dioxins. Quite bizarre. And ironically, they find oxygen toxic. We find their electron acceptor toxic. They find ours toxic too. For those of you who are into the science, here's the equation for that reaction. I've got on the right here just a little illustration of how these bacteria do this. It's quite well understood. The enzyme that catalyzes this transfer of electrons to carbon to chlorine bonds to release the chlorine atom are called reductive dehalogenases. And it's like molecular Russian dolls. So inside this enzyme, there's a cofactor, which is vitamin B12, one of nature's great works of art. Very similar in structure to heme or to chlorophyll. And inside the vitamin B12, instead of iron for heme or manganese for chlorophyll, is a cobalt atom, which acts as the active center of the enzyme. It's actually the cobalt atom that's delivering electrons to the carbon to chlorine bond. So we looked at the bacterial community to see if there were any of these organohalide respiring bacteria in the Harbour sediments. And we did indeed observe close relatives of known organohalide respiring bacteria. And so they were present at around 2.7% relative abundance in the sediments. And you might think, well, that doesn't sound like very much. But keeping in mind that there's around 500 million cells per gram of sediment in Sydney Harbour. So it's really quite an appreciable number of cells. I might skip that. So we set up an experiment to try and grow these bacteria in the laboratory. It was quite a big experiment. We set up little incubations from all 10 samples from the upper and the lower cores, and we amended them with things that make the bacteria happy. And we observed them on a monthly basis for about 2 years, and absolutely nothing happened. So that's part of the heartbreak in science. We do these experiments and sometimes nothing happens. At 60 incubations over 2 years that were down the toilet. And so we were unable to grow these microorganisms in the laboratory, at least on our first try. And so we did some work with one of the known dioxin degrading bacteria called the Helicocoides. This is a picture of Lorenz Adrian, who was instrumental in making that discovery. He literally wrote the textbook on organohalide respiring bacteria. And so we borrowed that strain from him, and we were able to show, as he had previously, that you can break down tetrachlorodibenzodioxin to dichlorodibenzodioxin. And so that's a possible candidate for introducing into the harbour sediments to actually accelerate degradation of dioxins in the sediments. But the question is, how is it really relevant? You know, is it active at marine salinity? What can we use as a reductant? We use something called titanium citrate in the lab. It's very finicky to produce, it's very expensive. You can't put that in the field. And how can we supply hydrogen in situ? And so we did some experiments with something called sulfidized nanoscale zero-valent iron and found that that serves as a great reductant and also as a source of hydrogen. So we were also able to show that the Helicocoides was active at marine salinity. It wasn't particularly fast, but it's not a deal breaker. So that's encouraging. I'm just mindful of the time, so I'm flicking through a few of these so we can have a chat afterwards if people have got questions. And this is data just showing that sulfidized nanoscale zero-valent iron, which is effectively iron sulfide, can serve as a reductant. It's just as good as titanium citrate and even better as some of the other options like sodium sulfide or iron sulfide. So that was positive news, and it's something we're thinking about including in our amendments into the harbour sediments, although the harbour sediments already have a fair amount of iron sulfide in them, so they might not need it. And this was a graph just showing that the sulfurized nanoscale zero-valent iron also serves as a perfectly fine supplier of hydrogen. So obviously pumping hydrogen gas into the harbour sediments is not something that we're interested in doing. But if we can put something in there that as it breaks down, it releases hydrogen. The other option is putting an organic substrate in and having that be fermented to produce hydrogen gas, and that's a standard approach to stimulating bioremediation processes in anaerobic environments. But we found that we can actually use a mineral source to supply hydrogen in situ in the harbour sediments. But we wanted to revisit our approach to growing dioxin degrading bacteria that were native to the harbour, not an imported strain. Somewhat reluctant to introduce an organism that's not from Australia into the harbour sediments. And so we came up with a strategy to actually enrich for organohalide respiring bacteria on a different substrate in the first place, and then test it if we saw activity, then test it to see if it could actually break down dioxins. And that strategy worked. So this data is showing that when we add in the laboratory to our flasks with sediment samples from the harbour, chlorinated compound called perchloroethene, this is actually a dry cleaning solvent, then we would see reductive dechlorination of the dry cleaning solvent. So the removal of chlorine atoms from what it's called perchloroethene, which is tetrachloroethene, down to trichloroethene with one chlorine atom removed, and then down to dichloroethene with 2 chlorine atoms removed. And we were really excited about that because it's the first time anybody's observed dechlorination activity in sediments from Sydney Harbour using the indigenous microflora of Sydney Harbour. So that was exciting, but the real test is ultimately, do they do that when you add dioxins? But before we did that, we actually looked at what sort of bacteria we were enriching in these sediment microcosms in the laboratory, because the bacteria, it's a respiratory process. So as they're degrading the pollutant, they are harvesting energy. So they're multiplying and growing and increasing in number. And this is something we exploit in biological remediation applications, this sort of snowballing effect. And what we saw was a large enrichment of this Dechlorination lineage, which we're pretty excited about. We did see it in the original sequencing, but it was at very low abundance, but we ended up with around 12% of the community being that organism. We also saw a massive enrichment of a different kind of bacterium. These are the sulfate-reducing bacteria. And what they're doing is respiring sulfate, which is quite abundant in marine environments. And so in these sediments, we would expect to have a lot of sulfate-reducing bacteria, and they're kind of, they're like arch enemies, the sulfate-reducing bacteria and the organohalide respiring bacteria. They've been enemies for over 1 billion years, still learning to live together. But it was encouraging, and we were a bit surprised. We kind of thought the Helicocoides would be enriched from the harbour sediments because it was also there. But under marine salinity, it seems like Dehalobium outcompetes Dehalococoides. So Dehalobium is like the marine version of Dehalococoides. So we then added tetrachlorodibenzodioxin, TCDD, and octachlorodibenzodioxin, the OCDD. That's the most toxic and the most abundant dioxins in Sydney Harbour sediments, to these microcosms to see if the bacteria could break these down. And after a very anxious 4 or 5 months wait, certainly anxious for a master's student who has a candidature of around 2 years, we started to see the liberation or the transformation of the parent compounds, the TCDD and OCDD, liberation of chlorine atoms from those compounds. So we saw tetrachlorodibenzodioxin transferred to trichlorodibenzodioxin, therefore a massive decrease in the toxicity of that compound. And we also saw octachlorodibenzodioxin transformed to heptachlorodibenzodioxin and then further to hexachlorodibenzodioxin, so the removal of 2 chlorine atoms. And so we've been monitoring these since then, and there's been additional dechlorination to pentachlorodibenzodioxin. And so we're really stoked, really excited about that result, that we actually, from bacteria that are in the harbour sediments, can dechlorinate these compounds that are in the harbour sediments and are a risk to human and environmental health, and the reason for the commercial fishing ban west of the Harbour Bridge. And so also, highlights that, this is the first ever octachlorodibenzodioxin dechlorinating culture that's never been observed before. And we also, interestingly, you might be wondering what's the difference between the red and the blue lines. So the red lines are the same as the blue lines, but with an amendment called lecithin. It's actually a food grade emulsifier or surfactant. And we added that because one of the limitations of dioxin degradation is that they're not very soluble in water, which means that the bacteria have a hard time getting at them. And so if we add a surfactant, then it makes the dioxins more biologically available. So we have a way of increasing the availability of the dioxins, or the access for the bacteria to the dioxins in the harbour sediments, with something that's benign environmentally. All right, so I'm going to wind up here with the last few slides. So these are the conclusions, just going back to our original 4 questions. Are dioxins still present at high concentrations? Yeah, they certainly are. There's still a risk to human and environmental health, and Homebush Bay continues to serve as a source zone, contaminating the rest of the harbour as sediments move around. Question number 2, is dioxin degradation occurring in Sydney Harbour sediments? So yes, for the first time we have observed evidence of that dioxin concentrations have dropped, and that's consistent with bacterial dechlorination activity. Are there dioxin degrading bacteria in the harbour? Yes, there are. Helicocoides is present, but Dihalobium is more abundant and seems to be the important one for dioxin degradation, and is most likely responsible for the dioxin degradation that's been observed in the Harbour sediments. And question number 4, can we grow them in the lab? And the answer is yes, we can now grow this Dihalobium from the Harbour in the laboratory. So what? Is that good? What's the significance? So these are our claims to fame from this study. The first evidence we have of dioxin biodegradation in Sydney Harbour. We have the first evidence that Dihalobium can degrade dioxins. Helicocoides has been seen to do that before, but Dihalobium has never been seen to do that. We have the first evidence that octochlorodibenzoedioxin can be biologically dechlorinated. That's quite exciting. The first evidence that sulfidized nanoscale zero-valent iron can support dechlorinating bacteria, and we're hoping to deploy that in the field. And the first evidence that lecithin enhances dioxin biodegradation. So what next? And we've done this a lot in the past with different compounds. We've been doing this for about 15 years. So we start with an inoculum from the environment. We generate enrichment cultures, and that's kind of the stage that we're up to. And then what we do is take those enrichment cultures to inoculate large-scale cultures in beer kegs, and then deliver those to different sites. So we've been doing that on land. This is the Altona Chemical Complex in Melbourne. In the middle, we have that's Southlands in Botany, and on the right, that's Deer Park in Melbourne. So all sites that have been contaminated with chlorinated compounds of different kinds, and that we've been growing bugs up and introducing those into the environment to accelerate the degradation of those chlorinated compounds in the environment. And so, as Hamish mentioned at the start of the talk, I established around a decade ago, and it was sort of reborn again in 2016, a company that's now known as Novorem, which makes all the expertise that we have at the University of New South Wales in the School of Civil and Environmental Engineering available to industry partners, or commercially available to people who are practicing contaminants. Should have these expertise at their fingertips. And that was something that was lacking in Australia, which I think was limiting the uptake of bioremediation. And so that company operates out of Mooresvale, south of Sydney. And it was the birth child of 2 companies. So Novorem was formed out of my original company, which was called Micronovo, and another one called Enrotech. And the company Micronovo still exists, but it no longer serves the bioremediation consulting industry. It's a publisher of kids' books and puzzles on microbiology. So if you're looking for presents for kids' birthday parties, endless birthday parties, and also for Christmas, then jump online, spend some money, and that will give me funds to pay another illustrator to make another book. That's my hobby. It's a bit bizarre, but there you have it. I'll finish up there. Thanks, guys.
Thank you so much, Mike. That was a fascinating, great talk. The questions are streaming in already, so for those of you who don't know, if you have a question, there's a Q&A button down the bottom of your screen. Just click on that and type your question in, and we'll try and get to it. Yeah, amazing stuff. I'm not sure whether to be encouraged or disheartened or all of the above, but yeah, thank you so much.
No, it's exciting.
It is. Yeah. Okay, let's get to the questions then. So Carl Ramirez asks, are people still being born with dioxin-related defects? If so, could Dihalobium or other dioxin-degrading bacteria be used as a probiotic to prevent these birth defects in contaminated areas?
Yeah, that's an interesting question. So my understanding is that they can, some of these defects can be intergenerational. So I think in Vietnam, people are still being born with birth defects, and probably other people around the world as well, where there's high dioxin concentrations, although Sydney Harbour's one of the 3 most contaminated harbours in the world. So could they be used as a probiotic? I think it probably makes more sense. I think Dihalobium, maybe not Dihalobium because it's a marine organism, but possibly the Helicocoides could survive in a human. The gastrointestinal tract, heavily colonized by microbes, is an anaerobic environment. So it's possible. But I think it's a more sensible focus, or more rational focus, to be cleaning up the environment and preventing the exposure in the first place. Thanks, Carl. And hi, Carl.
Thank you.
I know Carl.
Okay, excellent. Good. It's all about community here at Science at the Local. Riley Ferguson has a question. Can these bacteria have off-target effects? How do you test or predict what other compounds they might degrade?
Yeah, I mean, really the only way to do that is to test them one by one. So in terms of off-target effects, I guess these might be positive or negative. So, you know, it wouldn't surprise me if this Dehalobium culture turns out to be able to degrade other pollutants that are in the harbour sediments, other chlorinated compounds like the dibenzofurans, which I haven't spoken about today, and beyond octochlorobenzodioxin, hepta, hexa, tetra. So it wouldn't surprise me if they're degrading other things. But it's on the negative side, microorganisms can transform pollutants into compounds that are actually more toxic than the parent compound. So a classic example of that is perchloroethene degradation. I kind of illustrated the steps in that, but it goes from trichloro to dichloro to vinyl chloride and then to ethene, and ethene is harmless, but vinyl chloride, the second last step, is the most, it's not the most toxic, but it's the most carcinogenic of those, all the compounds in those steps. So it's also possible that the octochloro or higher chlorinated dioxins are broken down into tetrachlorodibenzodioxin, which is the most toxic of all of them. And that's certainly something that we're testing in the lab. We haven't seen it happen yet. And we also haven't, so with the transformation of octochlorobenzodioxin to pentachlorodibenzodioxin, we don't actually know, because we don't have the standards to figure it out, what the chlorine atom arrangement around the dibenzodioxin organic backbone is. And so if that chlorine arrangement can result in the 2,3,7,8 tetrachlorodibenzodioxin production, then that might be a problem. And it's something we certainly need to nail down before we hit the field and start introducing these organisms, or increasing the abundance of these organisms artificially in the environment.
Okay.
Thank you. Next question, Kate Foster asks, do we know where all the dioxins in the water have come from? Burning biomass smoke from wood heaters, controlled burns and bushfires put dioxins into the air. Does some of this get into the water?
Yeah, that's a good question. And we don't really have an understanding. So even some of the, you know, really far up tendrils of Sydney Harbour or Port Jackson estuary have elevated dioxin concentrations. It may be a consequence of bushfires. It's surprising somehow that the dioxin contamination from Homebush Bay has got that far, but certainly even the cleaner bits of the harbour still have elevated dioxin concentrations. It might just be an Australian thing.
Next question is from Rob Agars. Could you comment on the problem of PFAS contamination?
Sure, it's a big problem. That's a really tough one. So again, PFAS, or per and polyfluorinated alkyl substances. You know, these are also halogenated organics like dioxins, but instead of chlorine atoms, we have fluorine atoms. Again, this is an umbrella term for around 4,000 different chemicals, and they have been dispersed widely through the application of AFFFs, or firefighting foams. And for decades, people believed that these things didn't have negative impacts on human or environmental health, but there's more and more evidence that they are having, such that some of these compounds, out of the 4,000 at the moment in Australia, 3 of these are now regulated. That's PFOS, PFOA, and PFHXS. And these are extremely recalcitrant. So dioxins are hard to break down. Those 3 compounds are even harder. And so we've been working on that for a number of years now. without any success to date. So we've been able to break down other compounds in the PFAS mix, some of the most abundant ones in firefighting foams. So we've made some useful discoveries, but those three are hard nuts to crack. And part of the reason for that is that there's very few natural products in the environment that are fluorinated. It's a really rare thing to see naturally. And so microorganisms haven't really been exposed to fluorinated compounds. The carbon to fluorine bond is a very strong bond. It's not so strong that it can't be broken by microorganisms, but because of the strength of that bond, it requires a lot of what we call, I'm going to call it grunt, it's reducing power. And so it actually doesn't make sense theoretically that a bacterium would use a fluorinated compound as an electron acceptor for respiration, like they do the chlorinated compounds. So that's a big problem. And I think the solution is probably going to end up being locking those compounds up and reducing exposure in perpetuity, or using fairly aggressive chemical approaches to break them down, or using physical approaches to break them down. Or potentially combinations of chemical and biological processes to break them down. That's something we've been looking at. But you're right, it's a massive problem. If people have been hearing about the Westgate Tunnel palaver in Melbourne, that's PFAS. Williamtown, north of Newcastle, that's PFAS. It's a major problem. I should add to that, the perfluorinated compounds, whilst there are impacts on human and environmental health, they're not nearly as gnarly as something like tetrachlorobenzoedioxin or other chlorinated solvents, which are highly carcinogenic and very toxic. So PFAS are relatively benign, but still not completely harmless.
Okay. Next question is from Annie Panesian. Was there anything special about the HBB4 sample that could explain the very high concentration of dioxins in that sample, especially compared to the other samples, including the other HBB samples?
Yeah, Stuart Taylor knew it was there. So what was done, so land was reclaimed with dioxin contaminated waste, and that was, so there was really, really high concentrations in certain areas, just right off the edge, the eastern side of Homebush Bay, right off the edge of the Rhodes Peninsula. And so to about a depth of 0.5 meter, I think, about 30,000 cubic meters of sediment were removed. That happened around 2010. And the dioxins were thermally dissolved from the sediments and then destroyed in a thermal process. But there are still patches in Homebush Bay, hotspots, where there are elevated concentrations. And if you go down under the area that was excavated, because they basically put a cloth down to stabilize the sediments and then covered it in gravel. If you go down below that, there's still really high concentrations of dioxins under that. And so it's, like contaminants always in the environment, they're heterogeneously distributed. And so there are patches that are really quite high concentrations, and then there are patches that actually seem to be okay. And the ones that are okay, I think, are mostly sediments that have been brought down by the creek that flows into Homebush Bay. But yeah, there's hotspots for sure.
There's another question from Tom Jeffries, who says, "Nice talk, Mike. How are new tools like next generation," and hi Tom, like next generation DNA sequencing changing our way of monitoring the microbial response to contamination?
Yeah, well, it's a revolution. So from the 80s, when people first started sequencing DNA from the environment, and microbiologists had always been using microscopes and Petri dishes, agar plates to look at microorganisms. When they first started sequencing in the environment, there was this realization that, all these strange DNA sequences that no one had ever seen before. It's like, oh dear, our radar, our Petri dish and microscope radar is actually pretty limited, and we're only observing somewhere between 90 and 90 or 1 to 10% of the microorganisms out there. So exciting time to be a microbiologist over the last 30 years to realize that 90 to 99% of the picture was missing. And so with my the foundation of the company Novorem, the purpose of that was to put some of these environmental molecular tools, environmental diagnostic tools, into the hands of people that could use them. And so people now, when they're trying to characterize a contaminated site, which is what you have to do before you clean it up, not only are they looking at what contaminants are there and where they are and what concentration, but they're also looking at what bugs are there and can they break them down. Do they have the genes encoding the enzymes that can break those pollutants down? And so people are using molecular tools like DNA sequencing and quantitative PCR routinely now to characterize a site, and then if there's some kind of manipulation of the site to enhance the biodegradation processes, to see how that's going. Are you seeing an increase in the abundance of the microbes that are responsible for these the contaminant degradation, as you would hope to be seeing?
Okay.
You go.
Oh, sorry. Next question from Aidan Pryor. Hi, Aidan. Do you have any advice for someone who might want to go into bioremediation as a field?
On the research side or as a practitioner?
I'm going to say as a practitioner, because I think I know Aidan.
Right. Yeah, so there's increasing number of environmental consultancies that are practicing bioremediation in Australia. I'm not going to name any of them, but yeah, there's plenty of them out there. I can certainly put you in touch with people. So my efforts outside of academia to try and get these tools into the hands of people who can use them, I've sort of generated a large network in industry, and some of those people come back, or those organizations come back to work with me in an academic context as a partner organization in something like an Australian Research Council linkage project. That's kind of my modus operandi at university. But there's certainly a large community out there and an increasing appetite to engage in bioremediation, because bioremediation as a remediation optional technology approach has fantastic credentials in terms of cost. It's much cheaper than a chemical or a physical approach, but also in terms of sustainability, again on cost, but also social sustainability and environmental sustainability, much superior credentials than traditional technologies. And I think, Australia's kind of been a bit slow to take it up, because it's hard to imagine that if something doesn't involve a really large machine, then it's not going to work. It's hard to imagine that these tiny little creatures can do this, but it's by virtue of their abundance that they can. Something to do with our mining and agricultural heritage with large machines. But there's also societies. So the Australasian Land and Groundwater Association is certainly a meeting place for a lot of people who are interested in bioremediation, and I sit on some panels there, the Sustainable Remediation Forum within ALGA, Australasian Land and Groundwater Association. And I sit on that panel actually with a guy named John Hunt, who was responsible for the excavation of the sediments in Homebush Bay with Tees at the time. So getting involved with ALGA, or by all means get in touch with me, and I can put you into contact with people who are practicing bioremediation. If it's on the academic side, then get in touch with me again.
Thank you. Another question from Rung Jen Wong. How fast are the dioxins being degraded, and how much faster do they need to be for industrial applications?
So what we've seen in the laboratory, these are ideal conditions, but the rates were really quite extraordinary. So if we could get those rates in Homebush Bay, then the whole thing would be done in a year. But it's wishful thinking to think that you can get those kind of rates happening in the harbour sediments. We're certainly going to be trying that in the coming years. But even if it's a fraction of that, then we can solve this problem within the space of 5 years. It's doable. And that's what's really come out of that research, is that it's like, oh, actually, maybe we don't have to put up with this for the rest of our lives.
That'd be amazing.
And our children's lives and our grandchildren's lives.
I'm going to sneak a question in, but if anyone else has any, please type it into the Q&A. Is there plans to revisit the site, in 2027 or some other time down the track to see if that was a real trend or a spike in degradation or ongoing monitoring?
Yeah, I mean, we're convinced by the trend because it was observed over multiple locations in Homebush Bay. So we definitely think that result is real. But we don't have any plans to survey for surveying's sake. We've got more active plans. So we are currently collaborating with an industry partner, Transport for New South Wales, to put together an Australian Research Council linkage project application secure funding to actually do the legwork that's required to do a trial in Homebush Bay. So we've selected a site where the dioxin concentrations are very high, and we're developing protocols for introducing the bacteria, a surfactant like lecithin and the nanoscale zero-valent iron into the harbour sediments and actually see if we can make it work. And so as part of that, certainly in the area where we're going to be running that trial, we'll be characterizing that site and doing the measurements again and making sure we know exactly the state of that environment before we manipulate it, such that we can be convinced that something that we did actually led to an acceleration in dioxin degradation. And I'm confident that we are going to see an acceleration in degradation. The question is whether it's going to be fast enough, to roll that out at a larger scale throughout Homebush Bay. But ultimately, if you can clean up Homebush Bay, you stop contaminating the rest of the harbour.
Can I
so we'll have to invite you back for a talk in a couple of years when you have those results.
Yeah, absolutely.
Sorry,
I
Kevin,
might sneak one in. Also, Mike, just a question. Towards the end of your presentation, you had a couple of pictures of culture delivery. I was just kind of wondering what does that actually entail, and just generally what is that process of culture delivery?
Yeah, so I can go back to that, I think. Can I? Oh, yeah. So on land, we're working with groundwater in these images here, and you can see these little pipes sticking out of the ground. And so they are pipes that are installed in boreholes that have been drilled down through the ground to hit the groundwater, the aquifer. And so what we do, you can also see these large like cubic meter boxes. These are called IBCs, or intermediate bulk containers. And so we draw up around 800 litres of groundwater from the aquifer. So this is contaminated groundwater. And then into that IBC, we add culture. And we add things that the culture likes, so often a food source. Sometimes it's lactate. This is a fermentable substrate that provides the hydrogen and the acetate that the bacteria need to, that's their Vegemite toast. Sometimes we manipulate the pH. If the aquifer is, more than 2 pH units away from neutral, then we'll try and drag it back to neutral just to give the bacteria a fighting chance in the first place. And then we just gravity feed it back into the well. We mix it in those tanks. These bacteria, as I mentioned, find oxygen toxic, so we need to protect them from oxygen. We mix it in those tanks and get that amendment mix all homogeneous, and then just gravity feed it back into the ground. And that inoculates the ground. So that's the probiotic for the polluted plumbing of the planet. It's reasonably straightforward. And the groundwater is moving. With a lot of these compounds, you have a source zone where the contamination actually occurred, but then the groundwater is washing over that and slowly dissolving it. And the idea is to get the bacteria into that slipstream so that you have the bacteria in the same locations as the contaminants. But yeah, it's not rocket science, and it's loads of fun. I'm a country boy, so I like being outside.
Yeah, looks like fun.
Okay, well, look, we might wrap it up. We've just ticked over the hour mark. So look, thank you so much, Mike, for joining us today. It was a great talk, and thanks to everyone for tuning in. As Kevin mentioned, we are definitely going to be back next year and really itching to be there in person. So please stay tuned to our channels of communication, email and Facebook and our website. And yeah, we'll be in touch soon. Possibly March is when we usually have our first event, but yeah, we'll be in touch. We're actually doing another little exercise where we're revisiting some of our past speakers. I was amazed to see we've had something like 70 come through since we started in 2014. So we're going to do a little bit of a where are they now and say hi to them and see what they're up to. So stay tuned for that too.
Yeah. Thanks, everyone.
Enjoy the rest of your Sunday.
Thanks, guys. Thanks,
Mike.
Thanks for the opportunity.
Thanks, Mike.
Cheers. Bye.
Bye, everyone.